The DNA of two parents with the healthy mitochondria DNA of a female donor. Mitochondrial DNA makes up 0.1% of a cell's total DNA. Part of the energy generator of the cell, it's passed from mother to child.
The donor's nucleus is taken out of the embryo, leaving behind healthy mitochondria. The nucleus is then extracted from the parent's embryo and added to the donor's embryo, which is then implanted in the womb.
Scientists say the nuclear DNA remains untouched, so things such as eye color, hair color, skin tone, and intelligence remain unaffected. But ethical questions remain, and church leaders oppose the process because an embryo is destroyed. They also fear this could open the door to more legislation that would allow for so-called designer babies.
Once you cross that line, it's very hard, both logically and in practical terms, to then not take the next step and the next step, and you end up in that future of designer babies, which everybody says they want to avoid.
In our morning rounds, outrage over the latest move toward designer babies. For the first time, Chinese scientists use new technology to alter DNA in human embryos. The experiment could eventually help change genetic code for generations. Eric, this is so fascinating. First, explain what the Chinese did.
Yeah, very fascinating. So what the Chinese did is they took human embryos that harbored a mutation that caused a certain blood disorder, and they literally went in and very precisely cut out the bad piece and replaced it with a good piece to eliminate that particular disorder.
This is a single gene mutation. So what other single gene mutations that are common could this be used on? Huntington's disease.
Yeah, there's literally hundreds of different diseases to which this could be applied. Think of things like Huntington's and cystic fibrosis and Tay-Sachs and sickle cell anemia, all of those.
BRCA1, the breast cancer gene.
Even the BRCA genes as well. The number one concern with this type of technology is you're changing what we call the germline. And those changes you make to the germline get passed on for all time. As long as that lineage exists, those changes are going to be propagated through to their children, the children's children, and so on. And we don't really understand yet enough of the genome, to be making certain types of changes. This technology also is not so specific. Other mutations are introduced at this stage when you make those changes.